So if a cell membrane was only made of phospholipids, the cell wouldn't be able to do very much because nothing can get in or out. And that's, again, good for defining the boundary of the cell and making sure the insides stay separate from the outsides. But there are important molecules, like food, for example, that need to get in, and waste products that need to get out. And mostly those are large or polar molecules or ions in some cases that cannot cross the membrane on their own. There are some important things like oxygen. Oxygen gas is just a small nonpolar molecule, and so it easily crosses the membrane, as does carbon dioxide. But everything else cannot cross the membrane on its own. So to get other things in or out of the cell, you need proteins. And proteins are, in fact, one of the, if not the most important component of a cell, because pretty much everything a cell does, whether it's at the membrane or in the nucleus or any other part, requires a protein in one way or another. So proteins are, as we'll learn, are involved in controlling the membrane potential and in signaling between cells, but pretty much everything else, every enzyme, every reaction that is carried out in a cell involves a protein in some way or another. Also, as we'll see, as we've already seen, proteins are really important for the structure of the cell. So you may already know that proteins are made of these subunits called amino acids. So an amino acid is a molecule like this where you have a central carbon surrounded by another carbon, or attached to another carbon, which is part of this carboxylate group, so a carbon with two oxygens attached, and then an amino group, which is a nitrogen and two hydrogens. In fact, this is where the name amino acid comes from, because this group is called an amino group, and this group is a carboxylic acid. And so that's why we call it an amino acid. And then also attached to that central carbon, there's a hydrogen, and then another group called the residue, which is what the R stands for, sometimes called the side chain or the side group, or just the R group in some cases. And that is the kind of important part of the amino acid. It's what gives the amino acid its properties. There are 20 different side chains that make up the amino acids that are used to make proteins in our cells. Each one is slightly different. They differ in size and shape. Some are polar, again, so there are polar amino acids, or amino acids with polar side chains. There are nonpolar ones, meaning they're not attracted to water. Some are ionizable. That means that if they gain or lose a proton through an acid-base reaction, they can have a positive or negative charge, so their charge can depend on pH. And it's these different side groups that give the amino acids their properties, which in turn gives the proteins they make up their properties. And to make a protein, you attach two or more amino acids together by this special bond called a peptide bond. So this is two amino acids here, and you make a peptide bond by removing an OH from the carboxylate group or carboxylic acid group of one amino acid and the hydrogen from the amino group of another, and you form this bond here where you have a carbon double bonded to an oxygen, and then single bonded to that amino group. And this, in fact, this is what the peptide group actually looks like. It forms this characteristic kind of zigzag shape, and this is the backbone of the amino acid. It's what of the protein. It's what holds all the amino acids together, and it gives it its structure, its shape, its strength. And it is important because you notice the R groups, the side chains, are not involved in the peptide group. That means they kind of stick out. And so the R groups then are free to interact with each other and interact with other molecules and give the protein its properties. In fact, the peptide bond is sometimes used to describe proteins. So you may sometimes refer or see a protein referred to as a polypeptide, and it basically means the same thing. So the word polypeptide and the word protein sometimes get used interchangeably, but really a polypeptide just refers to a chain of amino acids linked by peptide bonds. And so we can talk about the structure of a protein at 4 different levels. The primary structure of a protein is just the sequence of amino acids that makes it up. So every protein has a unique primary structure. That primary structure, of course, is determined by the nucleic acid sequence of the gene that encodes the protein. So every amino acid has what we call a 3-base codon that encodes it, and that is where that is found in the gene sequence for that protein. And then the secondary structure of a protein is what happens when the protein starts to fold up on itself. And there's different types of secondary structures. This alpha helix is a really common shape. What's important about the secondary structure is that it's formed entirely from interactions between groups that are part of the peptide bond itself. So if we go back here and look at the peptide group, you notice there's this oxygen here and this nitrogen with the hydrogen facing down. And so different peptide groups from different parts of the same protein can interact through those groups. And that's what gives rise to and stabilizes that secondary structure. And importantly, the primer, or the amino acid sequence, is not involved in the secondary structure as much. So the amino acids on an alpha helix like this, for example, are not part of the alpha helix itself. I mean, the side chains, they sort of stick out from the alpha helix like little spokes on a wheel. And so that means that they are then free to interact with each other and interact with other parts of the protein to produce tertiary structures. So tertiary structure is the sort of next level of folding, where you have the side chains here. Again, we have 4 alpha helices in this one polypeptide, and they're all kind of attracted to each other because of their side groups. And so this is the sort of highest level of structure that you can have for a single polypeptide. But many proteins are actually consist of more than one polypeptide that are bound together in what's called quaternary structure. So a lot of proteins, especially membrane proteins, like some of the ion channels and receptors we're going to talk about, are polypeptide, are multi-subunit proteins. And so each subunit is its own separate polypeptide. Sometimes you have subunits or proteins where each subunit is the same. So it may be each of these. In this case, we have 5 subunits in this complex, and they could all be the same or they could all be different. And sometimes quaternary structure is permanent. Sometimes proteins undergo changes where the subunits may dissociate. They may take on new subunits. And in fact, it's that change in shape that makes proteins, allows proteins to do what they do. So the shape, though, is determined either directly or indirectly by that primary structure. So if you change the primary structure, meaning if you change the amino acid sequence of a protein by changing the DNA sequence of its gene, for example, through a mutation, then you can change its structure, and that structure will change its properties. So for example, if you have a membrane protein like this, which is what we're interested in today, these membrane proteins, or any protein in the membrane, does so because it usually has amino acids, like in this part of the protein, for example, that are inside the membrane. And of course, they're going to find themselves inside the membrane, or prefer to be inside the membrane, usually because they have non-polar side chains. So those non-polar side chains are what keep these amino acids inside the membrane, and that's what keeps the protein inside the membrane. So again, if you change the amino acid sequence, so if you were to have a mutation, for example, where one of these amino acids in this part of the protein became polar, for example, then that protein would change shape. It would perhaps not as fit well in the membrane, and it would not work. But again, so the shape determines its function, where it ends up, and then its ability to change shape, as we will see, also is important for how proteins behave. So like I said, we're mostly interested in membrane proteins. So that's a protein that's either embedded in the membrane or passes through the membrane of the cell. And it's these proteins that are especially important because they allow the cell to communicate with other cells and with the fluid surrounding the cell. So an important type of membrane protein are ion channels. These are proteins that sit in the membrane, and typically they consist of multiple subunits. So like this one, for example, has, again, 5 subunits, 5 different polypeptides that make it up. And importantly, they are arranged in this kind of barrel shape surrounding kind of a central pore. And that pore then will be a place where ions perhaps can go in or out of the cell. So for example, and again, because of the property of the amino acids, the protein is able to take on this shape. So for example, if this is sitting in the membrane, then presumably The amino acids here deep inside the protein and deep inside the membrane would be nonpolar. And then the ones sticking out here in the water of the extracellular fluid or down here in the water that's in the cytosol would be polar. And then if this is an ion channel, then the pore, the hole that ions go through, will again let ions through, but not just any ion. So most ion channels are selective or have what's called selective permeability. So permeability just means the ability to let stuff go through. So an ion channel typically can be described by the type of ions it allows through. So let's say this is a sodium ion. That means that it will let sodium go through the membrane, through the pore of the channel, but not other ions like, say, chloride. The reason it lets sodium through and not chloride is presumably on the inside of that pore there are amino acids which maybe are negatively charged and therefore will be attracted to sodium but might repel a chloride ion. And so those sodium ions then can enter the pore and then eventually cross through the channel to the other side. In this case, from the extracellular fluid to the cytosol. Although importantly, ion channels are not particular about the direction that ions go. So they're permeable for the type of ion, but they're not going to decide which direction the ion moves. Most channels will let ions go either direction, in or out of the cell, and we'll learn later on what determines which direction the ion moves. And so ion channels, some are what we call constitutively open, or another name for that is a leak channel. That means that they're just open all the time. They don't ever close. And whatever ion is selected for this channel, if it's present, it will go through. And then, but many ion channels are what we call gated ion channels, which just means they can be in at least one of two states, either open or closed. And some sort of signal has to trigger the channel to switch from one state to another. A really important type of gated ion channel, which we'll talk more about later, are these. They're called voltage-gated ion channels. And a voltage-gated ion channel is open or closed depending on the membrane potential or the membrane voltage. And we'll learn all about membrane potential later on. But for now, we'll just think of it as the difference in electrical charge across the membrane. So over here, in this case, this ion channel is closed because there is a negative membrane potential. And that means that on this side of the membrane, on the inside, there is a net negative charge. So presumably on the outside, there would be a net positive charge. And we can measure that difference in charge as voltage. And here we say it's at negative 65 millivolts. So that means that's just a measurement of how negative the inside is to the outside. And that large net negative charge will pull positive charges in. So these voltage gauge channels have some positively charged amino acids close to the pore. They get kind of pulled into the cell when it's negative, and that causes the channel to be closed. But then if the membrane becomes more positive, so here less negative, I should say. So it's still negative, but not as negative. So the net difference in negative charges on the inside versus outside is smaller. So, still negative, but not as negative. So we're closer to, we've gone from negative 65 to minus 40 millivolts. There's less inward pull on those positively charged amino acids, so they move out of the way, and that causes the rest of the protein to change shape such that the pore opens. So that's why we call it a voltage-gated channels. We'll talk about these more actually in the next chapter when we talk about action potentials. And then another really important type of ion channel are called ligand-gated ion channels. So a ligand is basically just something that binds to a protein. And so proteins, in fact, most of the time, they do what they do by binding to other molecules. And that might be other proteins, or it can be other types of molecules. And ion channels are just special proteins that bind to some sort of molecule, usually on the outside of the cell. And that triggers them to either open or close, depending on the type of channel and the type of ligand. And these are going to come up, are going to be important later when we talk about neurotransmitter receptors, because many of the important neurotransmitter receptors are ligand-gated ion channels. So this, for example, is a picture of the GABA-gated chloride channel. And so this is a GABA receptor. GABA is a neurotransmitter, which we'll talk about later. It stands for gamma aminobutyric acid. And the GABA receptor has a binding site for GABA. That means this little pit here is just the right shape for a molecule of GABA. When GABA binds to it, it causes the rest of the protein to change shape and open, and then the pore that it creates when it's open is permeable to chloride ions, and therefore it allows chloride to go through. And so we'll talk about lots of examples of this, and we'll show that not only are these sensitive to their neurotransmitter, they can be sensitive to other molecules as well. So some ion channels can be both voltage-gated and ligand-gated. This is a channel for this is called the NMDA receptor. Actually, it's a receptor for the neurotransmitter glutamate, but we call it the NMDA receptor for reasons we'll get into later. And so that means it is sensitive to glutamate. Glutamate is its ligand. If glutamate is present, it causes the channel to open. But it also has this little magnesium ion that kind of sits in here and blocks it. And that magnesium ion will only leave if the membrane potential becomes less negative. So at minus 65, it sits here and blocks the channel. At minus 30, it leaves. And that, again, as long as glutamate is also present, it will let ions go through. So you can have ion channels like this that have multiple triggers. The last important type of membrane protein to be aware of are ion pumps. So these are proteins that sit in the membrane. They allow ions to go through, but unlike ion channels, they are picky about direction. So again, ion channels are passive. They're either open or closed, but if they're open and the ions that they're selected to are present, they can let them go either direction. Ion pumps, on the other hand, really only move ions in one direction, and they are not passive. In fact, they require energy. This is a really important example of a ion pump. It's called a sodium-potassium pump, sometimes called sodium-potassium ATPase, because it uses ATP and uses the energy from ATP to move ions. So in this case, what it does is move sodium ions outside of the cell. So these sodium ions are moving from the cytosol to the extracellular fluid. It has, in fact, 3 sodium binding sites. So if sodium is present on the inside of the cell and ATP is present also, then the cell converts ATP into ADP, uses that energy to let sodium go through. And then it also has then binding sites for potassium that pull potassium into the cell from the extracellular fluid. And so it forces them to go, forces sodium out of the cell and potassium into the cell using ATP, again, regardless of the actual concentration of those ions. All right, in fact, next time we will talk about the movement of ions across the membrane.